connection with this chapter. It's still under the genetics unit, but it's population genetics. And so they're together right now. So I'm sure people argue, oh no, we should start this as the evolution unit, or no, it's still the genetics unit. Okay, so the second one won the fight in this book. In my other book, it's in the evolution unit. Either way, I just bring that up so you guys see this huge relationship here, and it's a great transition from us finishing up like typical genetics, Mendelian genetics, and then moving on to evolution. You should be able to see this bridge, right? Because genes, right, we're studying how genes are inherited for genetics. Now we're studying, when we get to evolution, how genes, the abundance of genes, the frequencies of different genes, really, different alleles, I want to call them, then is evolution. Because you guys need to realize evolution is simply changes within populations. Okay? Populations evolved. Individuals do not evolve. You do not evolve. Okay? The human population evolves. Pea plants evolve. Okay? So there's really, at this moment, there's nothing to be controversy. There's nothing to be debated. Now, we'll get to that. Don't worry. But right now, we're just talking about the fact, and it's a fact, that populations change. Right? Some alleles get more common in a population. Like in the human population, the dark hair allele is more common than it used to be. You guys with me on that? Can you guys, right? That's a fact, right? You guys see what I'm trying to point out here? So, again, we're studying that. Mr. Allen, focus for me, okay? So, evolution is defined as a change in populations through time. Also, many times this definition is over generations. Okay, it's saying the same thing, basically. There's two time scales. Now, the one we're going to concentrate on right now is this first one, which is what? Yeah, oops, I've gone crazy here. Yeah, it's micro, if they'll show it to us. And this is what I was just talking about. So change within species, which can occur over dozens or hundreds of generations. That we're going to consider small. Large changes, I'm going to mention it right now as a definition, and then we'll get to it on the next chapter, is macro. This is what we can discuss and debate if you'd like. And that is speciation, the origin of new species. Okay? And this takes lots and lots and lots of generations for it to happen. So it's going to happen quicker in organisms that reproduce faster. Okay? Okay, so now we do have, some new vocabulary for you maybe. You probably haven't thought of population genetics before, but it's basically the study of microevolution, that change within populations that we can observe, that we can study, and then remember that term I had you put on your learning plan, quantitative analysis. We can use numbers, we can do math in this type of field. This is probably why most students think that this is one of the hardest classes in their biology degree. I know it was for me when I got my BS, my Bachelor's of Science in Biology. I didn't even take population genetics. I stayed away from it. I took other genetics because that's one thing that's kind of cool when you guys pick your majors. But if you're in a good school, you have lots to choose from. I should say if you're in a good department. And so I just stayed away from this. This was too much math for me. But I did have to take it in my master's, and it was hard. It was hard. I got an 88.5% in that class and worked as hard as I could. It was okay, though. I did. It was okay. Like you guys, I was hoping maybe he'd put it up to an A. No, but it's okay. Okay. I know, everybody can dream, right? Okay, so now another word is gene pool. Listen to this.
The gene pool is a collection of all of the genes and their different alleles that are in a population. In simple population genetics models, the gene pool is imagined to be an infinitely large body of water containing all the alleles. For example, if a population of plants consists entirely of homozygous purple-flowered plants, then its gene pool has many copies of alleles that make purple flowers, and no alleles that make white flowers. In this case, the frequency of the allele for purple flowers is 100%, and the frequency of the allele that makes white flowers is 0%. If the population consists entirely of homozygous white-flowered plants, then the frequencies of alleles in the gene pool are reversed. 100% alleles for white flowers and 0% alleles for purple flowers. It is also possible that both types of alleles are present in one gene pool.
Okay, so I know it's small, but, I think, did he do pretty much half and half with that? So notice we're looking at just the allele here, not directly the phenotype. Directly, how many big P pieces of gene are in the population? How many little p genes or alleles are in the population? So they have a nice little animation here. That's called the gene pool. It's all the possibilities in a particular population. Right now, right here, if we got isolated, everybody else on Earth died except for us right here, you guys would be the gene pool. It's whatever genes we have to continue on the whole human Homo sapiens species. Okay? What's hidden in your DNA. Okay, let's listen to this one.
Homozygous purple-flowered plants are crossed with homozygous white-flowered plants to produce heterozygotes in the F1 generation. Note that the frequency of each allele is the same, 50%, in the F1 generation as it is in the P generation. When the heterozygotes in the F1 generation...
Okay, stop, because if I was to ask you allele frequencies of these purple flowers, you can't see the little p's. They're hidden behind, right, the dominant one. But did you hear them? So the population, if they were half white and half purple, just like that, being purebred, being homozygous, they have the same allele frequencies. You guys see what I'm talking about here? Okay, so we're getting at the hidden allele when we're studying population genetics, because even though it's not expressed, it's still in the gene pool.
Are crossed, the frequency of each allele remains the same.
So I just adjusted my sound, so if you guys happen to watch this, realize the beginning's probably awful, but I pulled it down now, and so now you should be able to hear me. So just get through that beginning part if it's really loud. Okay, just another vocab term, polymorphic. Poly means many. So here we're talking about, it's just two, not even many, two or more different alleles for a specific gene and for a trait. You got down to the third bullet, remember genetic variation is due to the sequences in the DNA, your nucleotides, and then also affecting the sequences of the amino acids. Remember, sexually reproducing organisms normally have high variation. Think back of the movie with those fish. And then the two sources, mutation and recombination of alleles during meiosis. Which paper?
The genomes from two different individuals are 99.9% the same. This means that the 3 billion base pair human genome is expected to contain about 3 million variable sites. One source of all that variation is mutation. Mutation gives rise to new alleles, such as when capital A mutates to lowercase a. Before mutation, only capital A alleles were present. The second source of variation is the recombination of alleles during meiosis. Crossing over generates new combinations of alleles at different genes. Before meiosis, only the combinations capital A lowercase b and lowercase a capital B occur. Crossing over between the two genes results in the recombinant types capital B and lowercase a lowercase b.
So this is one of your learning targets. You guys need to understand the source of genetic variation. So what's the first one up there? Mutation. Okay, the second one is, it's crossing over, and it does get a little more than that. Crossing over is the main component, but even back in just the segregation of the alleles through gamete formation, remember our square dancing little models, the randomness of fertilization. I would kind of, if we're going to do this, say that there's only two, I would say mutation, okay, is one of them. One's not more than the other. If anything, I would tend to think sexual reproduction is going to have more influence than mutation, because mutations are rare. But anyway, so it's sexual reproduction, but you need to understand what that means, which is crossing over, the segregation of the alleles randomly, right? And then the randomness of fertilization. Which sperm fertilized which egg, and what did it have in it? You guys with me on that? We've been talking about this for a while, okay? Okay, then, I didn't skip one, did I? No. Okay, then we get to Hardy-Weinberg. Now, this is the one where I really want to put your thinking caps on so you can so the lab's not as frustrating for you. Okay, first, Hardy-Weinberg, two different people. This is a model to figure out, it's a model to figure out when or at how much a population is not evolving. So hear me carefully with this. This has all sorts of issues with it. They're assumptions. You'll see them in a minute. They seem very unrealistic. They are. What they're getting at is they're trying to make a baseline for you. They're trying to make a zero when no population is changing. If we don't know what zero is, we can't figure out how much something is changing or how little something is changing. We don't have a zero or a baseline. Does that make some sense, you guys? Like, I tell you it's hotter today, but you didn't know what temperature it was yesterday. Does that make some sense? So this gives us a starting point or a comparison. And these mathematical wizards, figured this stuff out. Thankfully, we don't have to do all the crazy mathematics. We just need to utilize it. So these are the assumptions. There's a whole list I'm going to show you of what is necessary for it to truly be a baseline, for it to truly be a zero. First, this model only works with diploid organisms, okay, that have two of every allele. And when mating is occurring, there's no generation overlapping. So think about it with your pea plants. You had your parents. They made the first generation. The parents are removed from the situation. They are no longer available to give their pollen or their eggs. So no generation overlapping. We need a big size. We need a good size population for this to work. When we run our model next class, actually, Health Science 1 is going to be doing the simulation with us. It's cool for them to participate and learn about it also, but mostly just because we need more individuals. I want to, even still, doubling our size is still a pretty small population. But still, it's better than what we have just in this room at this moment. And the reason being is think about when you were flipping coins and stuff, you can get a run, right, where it's not showing 50-50 odds. Right? You can flip heads 5 times in a row. You're probably not going to flip heads 50 times in a row, though. You guys following me? Okay, mutation is negligible. We can't have any mutation. For example, when we do this, we're going to probably be working with A's. It doesn't matter. You got a big A and a little a. Okay? We can't have a big A mutate to a C or something else. That throws off the equation. We only have 2 different types of alleles. Reproduction is sexual. Gametes unite at random. This means one mate is not better than the other. This means reproduction is purely random. There's no sexual selection, for example. It isn't better for you to be a big A or a little a. Migration is negligible. We can have new people join us late. They can't come in and bring their alleles into our situation. Natural selection doesn't operate. I kind of already said this with the random mating. Big A is not better than little a. Okay, so that is all the assumptions. Can you see, is that practical? Is that realistic at all? What I just told you in the real world out there? No, but that's going to be when there's no evolution occurring. Okay, so now the equation itself. First, let's write it down. Do I have it in a few pages ahead? Do I have this whole equation? So write this down. P squared plus 2pq plus q squared equals 1. That's the Hardy-Weinberg equation. Now, there's another equation that's more even useful to start, and that is also P plus Q equals 1. Now, what are we talking about? Okay, think about this. The allele that says be purple versus the allele that says be white. There's nothing else. You're either a purple allele or you're a white allele. So in a population, we added up all the purple alleles, we added up all the white alleles, they should add up to what? And that's kind of hard to understand, because what if there's like 50 of purple alleles and 25 of white alleles? Okay, so hold on, but that's all there is. There's 50 and 25. So there's 75 total. So they need to add up to, if they're going to add up to 1, what are we talking about? The total. So let's talk about percentage for a second. Right? There's 50 and 25 together. What percent are they? I'm tricking you guys here. Okay, I have, how many alleles do I have total? No? How many alleles did I say total? 75. Okay, there's 75 alleles total. 75 I said of 50 I said of purple, 25 are white. So it's adding up to what percentage? 100. Now I did that messed up on purpose, but I want you guys to think about this. Okay? So when it says 1, it's talking about a fraction. So back on that the gene pool picture in the lecture, it said there was, this would be 0.5 for P plus 0.5 for Q, because they were half and half. Do you guys agree with that? 50% with this, 50% with this. That adds up to how many percent? 100%. Or if we're doing fractions and decimal, it adds up to 1. So I want you to be able to interchange between your fractions and your decimals rather quickly. And we'll practice doing that in a second when we do the example. So now real quick, look back up here. P. P is always going to stand for the big letter. I try not to use dominant as much as I used to, or recessive, because that also emphasizes that dominant's better than recessive, whether it's true or not. We do that in our brain. So just realize P is standing for the capital letter. Q is standing for the what? Right, the lowercase or small. So you guys understand how they're always going to add up to 1 if we're in decimals? Yeah? Okay, now, we are talking about the alleles when we use this frequency. The big piece or the little piece. And every organism has how many of them? Let's go with eye color for you guys. You have 2 alleles. One says be brown maybe, another one says be brown also. So how many do you have in your body? 2, because you are diploid. Okay? So now, what about the bigger equation? So this is just talking about the alleles. Each one. I have 2 in my body. You all have 2 of whatever specific allele we're talking about. But lots of times we have to determine it by looking at the phenotype. So we have 2 possible alleles in this case. How many different genotypes do we have? 3. P squared, 2pq, and q squared represent the alleles. So if the big P stands for a big A, this is big A and big A. So P squared is the mathematical equivalent of the homozygous big ones. What do you think 2PQ is? Yeah, because that Q is standing for the little one. And what do you think Q squared is? Okay? Now, thankfully, you're not ever going to solve for 1P or a Q from this big thing. But you guys will utilize this knowing that 2PQ stands for the frequency of heteros in a population. So back to our purple and white P plants. P squared is going to stand for the big B, big P in that case. Q squared is going to stand for, right, and we can get at them either direction. I can give you a phenotype, and you can tell me P's and Q's. I could give you a P, you can tell me Q, then you can plug them in. Okay, so I talked about that already, it being a baseline. Then, let's talk about 2 case studies. The peppered moss are the first one. So this is back in the 1800s. Okay, I'm switching gears. Stay with me here. We have a little story to hear. In the 1800s, when the Industrial Revolution was happening in England, they're a little bit ahead of us in our Industrial Revolution. Anyway, they're making soot and pollution, burning stuff. They're busy working like crazy. Meanwhile, they're hurting the forest around England, because England is huge, not England, London, England, okay? The forest around London is getting all sorts of pollution. So in your book, there's pictures, I'll show you in a second, but what is happening is natural selection is changing the frequencies in the gene pool, and it helps the population adapt to its changed environment. What am I talking about? The environment around England changed. There's these birch trees which are light, right? No, they're dark. They're not that light. They're kind of like this. And the lichens, these things that grow on the trees, we have lichens that grow around us too, but their forest, this was very common. The trees were covered with these lichens. And the lichens made the trees lighter. You could see to the A situation over there. They're hunted by birds. And so before the Industrial Revolution, the moth population, what do you guys think? Was it mostly dark or was it mostly light? It's mostly light. And you can't even see in this picture, it takes me a minute find it. I think it's right around here. There are 2 moths in each of these pictures to represent the camouflage. So the birds can't see the ones that are best adapted there, that have the genes that say be light. And that is because they're going to eat anybody that has any darkness. They're going to see them. They're going to have low fitness if they're dark in this case. Okay, the lichens die. They fall off the trees because of all the soot and ash and the pollution in the air. So now we have darker trees. What happens to the peppered moth population over generations? So now the ones that had the good gene, the good supposedly, right, allele, are now getting targeted. And the other ones are more camouflaged. I think it's right here. You can tell better in your book. Okay, and check this out. Now, so we saw evolution occur within the peppered moths. They were healthy, they had normal variation to start. But the alleles that said, be dark, became increased in the population when the trees got darker. We watched it reverse. They started cleaning up their pollution just like we did here, and the lichens grew back on the trees, and the population has shifted back to mostly being light. Okay, it's a very famous study. Now, your lab, the one you're working on right now, with sickle cell. So sickle cell is interesting. We noticed at first that there's a high frequency of sickle cell in Africa. And we were trying to figure out what is going on with these people. Why are they getting this disease? Now listen, remember that the sickle cell allele makes hemoglobin that's misshapen. You guys remember this from your reading? Okay? And so what happens is you get the sickle cell shape, and they're not going to work correctly. And it's also painful. If it's in a heterozygote, though, there's a dominant recessive factor, but it makes sense. They always do once you figure it out, why one is more dominant over the other. You're getting enough normal-shaped hemoglobin not to contort your blood cells. And if your blood cells are nicely shaped and you have at least 50% of the hemoglobin is holding onto the oxygen nicely, you don't get the disease. You guys with me on this? So it's very straightforward why it's, a recessive disorder. You need that all the hemoglobin must be messed up for it to really contort your blood cells. Now, there's one thing that's pretty useful for this. And in Africa, malaria, and actually around the tropic zone, around the entire Earth, malaria is more common. Okay? Malaria is a protozoan that attacks us. It kills more children than any other disease on Earth. So this is a very fatal, bad parasite to get. It comes from mosquitoes, but that's just the vector. It's a little protozoan, a little single-celled, animal-like organism that has to go through life cycles in mosquitoes and then in our body, or mammals in general can get malaria with these pretty red blood cells that we have like this. If they're heterozygous and half of their hemoglobin is kind of screwed up, the structure, I guess, is different enough. The malaria cannot complete its life cycle in these individuals. They are immune or resistant to malaria. So heterozygous individuals around malaria do not get malaria. Pretty cool? Okay? Now, that's a big environmental difference. Okay, we'll come back to it when we do the lab. Let me keep going. Some vocabulary. Gene flow, just like it sounds, flow, movement, is when, basically, organisms migrate. They move around. They change the gene pool. So back here, don't forget, you got natural selection. I'm going to talk more about genetic drift, gene flow, and mutation. These are all mechanisms that are going to affect your gene pool and what alleles are in your population. Okay, genetic drift is pure change by randomness. Now, we see it more in small populations. And an example here is genetic drift. One example is just random. So you have some flowers, some plants that have yellow flowers and white flowers. And this plant is just found in a particular region, let's say up in the northern plains. And the yellow is very rare. But here you have a population growing, and there's a good amount of yellow in this population. Okay, the good old buffalo are getting ready to migrate. They just happen to come down, right, and they're stepping on the plants, and they kill some of the plants. Randomly. They kill the 100 yellow plants and leave behind all the white. Did they change the gene pool? Was it calculated? Was it because of the environment? Was it because of sexual selection or anything like that? No, it was just total randomness. Okay? Now, if it's a really small population, there's probably better odds that, you know what I mean? If it's small, and then the buffalo come by, it's more chances of getting those few yellow in there. Okay, now, to memorize genetic drift with randomness, like things drifting around. Okay, founder effect here is just founding fathers, right? The idea of, colonists, right? Groups moving away from the original population. A small amount goes. There's going to be some genetic drift in there also. Whenever a group moves out, it's whatever genes they take with them. So that's called founder's effect. And that's what those circles in your plan and your learning guide had in them. This has happened with the Canada geese. So B over here, these are the geese that got separated. These are the founding geese that made it over to the Hawaiian Islands. And over many, many, many generations, they have gotten so different that they're not even the same species anymore. But they were Canadian geese when they first arrived, or Canada geese. Okay? So that's a founder's effect. Okay, the thing that increases with these situations where the population gets small is inbreeding. And inbreeding is going to lose diversity, right? Just like you saw with the little fish in the pools when we watched that, what's it called, the red queen effect. So another type is called a bottleneck, where a population goes down drastically. Again, whatever alleles are left there. This has happened with the cheetahs. It's happened with the California condors. The California condors were down to 20 individuals. They went and took them from the Sierras. They captured them. They took the last 20 condors. They split them up between San Diego Zoo and LA Zoo. We didn't want them all in one spot. What if something bad happened, right? They bred them. And over many years, I'm going to guess probably at least 15. You guys can look it up. This is a well-known study. They had enough individuals raised that they re-released. So there's free California condors. Still, they had inbreeding going on when we only came from 20 individuals alive. It was kind of cool, too. There's lots of neat stories. I have a book on it, and you guys are going to have an extra credit available, too, during spring break. But they puppet fed them, like with condor head puppets. They didn't want them to get used to humans at all, because the reason they're endangered was purely because of humans. They have enough habitat. It's hunting, and really just killing. You don't hunt. I mean, what are you going to do with a big, beautiful condor body? Right, I guess, but totally against the law. So lots of times it's just mean people. They can get hit very occasionally by cars, or they can also hit electrical lines, too. Nowadays, probably the windmills, but we didn't even have those back then like we do now. Okay, so this is called a population bottleneck. Just think down to the squeezing of the bottle, right, where you're getting a small amount. And this obviously is going to cause inbreeding depression, where the animals are not as healthy. Okay, I put this last quote here to, like I already talked about with genetic drift. Most evolutionary scientists and geneticists think that natural selection is what mostly affects changing populations. But there's a growing group of scientists that are like baloney. There's way more randomness to this than you're giving credit to. There's two scientists that debate a ton up at UNR on opposite sides. Okay, so one last slide here. Artificial selection, selective breeding, artificial breeding, whatever you want to call it, all means the same thing. We can do this. We can evolve organisms. We have been doing it for thousands of years. So these three guys are on the outside. We have bred them from rock doves. Same thing with our dogs. So rock doves are just what we call pigeons. And next time you're around a bunch of pigeons, check them out. Look at them. Some will have little flipped up tails. Some will have little flipped up neck feathers like this guy up here. Some have, the crop, I guess they extended. I haven't paid attention to that with the pigeons, but look for it if you want. So we want a fantail. We find one with the biggest flipped up tail, right? We breed it to another one with the biggest flipped up tail. Their babies have a little bit more flipped up tails. We keep going. There's lots of inbreeding in this, right? But we change species through natural selection, through artificial selection, just like nature does it naturally. Okay? Questions? Okay, I want you to do this, and I want you to write it down, because I want to see how we did. Do.